Welcome to Chapter 4, Sensation and Perception. After 2 chapters of boring technical stuff, we are now treated to exciting technical stuff. Slide 2. Can anyone see a dog in this? Slide 3. What about now? Slide 4. This one has me flip-flopping between 2 different versions of a 3D box. Slide 5, this one, and slide 6, this one. Slide 7. Here are 3 lines. Would you believe if I told you that they are the same length? Slide 8. What about now? Slide 9. In viewing this stimulus, people invariably perceive the diamond on the left to be darker than the diamond on the right, even though they're both the same color spectrally, just because the background colors give this deception. This also works with regular colors. Slide 10. This is the same kind of effect, but instead of being isolated to brightness, it actually uses colors. So the 2 central targets here are identical. Slide 11. This one's a little bit more brainy. Got a Rubik's Cube looking thing here. When we look at these pictures, our brain understands that it's the same picture of the same cube with the same colors with 2 different filters. But when we drill deeper into the experience, we should question why our brain understands this. Almost none of the colors are the same between the pictures, and the ones that are the same aren't perceived that way. Allow me to demonstrate. Slide 12. As you can see on the bottom, the patches that are colored orange on the left are perceived as red. But red is also perceived on the right by colors that are actually purple. But again, because of the context cues, we are perceiving these orange dots on the left as red and these purple dots on the right as red. Slide 13. Further, we can see that colors that we are perceiving as different, like blue on the left versus yellow on the right, are actually spectrally the same exact color. They're both gray, but again, based on the cues of the colors around it, we perceive gray as blue in the picture to the left and gray as yellow on the picture to the right. Slide 14. This one's a classic, believe it or not. Squares A and B are the same color here. The one that is in the shade and the one that is out of the shade. And slide 15 demonstrates this. Believe it or not. Slide 16. Let me start by saying, and I hope those demonstrations hint at this fact, we are not in direct contact with the external world. But, you may protest, everything about reality and our own perceptual sensory systems confirm that we are in direct contact with it. Remember, philosophers like Plato and René Descartes thought sense data was deceptive and therefore could not be used as a reliable source of knowledge. I posit that our brain and reality are working together to fool us. In both a sensory and perceptual way, we are merely picking up on cues from the external world and cobbling together or constructing a partial model of reality. But fear not. Our internal model of reality, even though it is partial, it will suffice. That is, it works, and it allows us to get along with each other. The bottom line for humans is that our internal models are tuned by evolution. Our brain is a prediction and survival machine, not an instrument to accurately measure external reality. That is, the palette of colors, sounds, tastes, and smells that we can perceive are fine-tuned to the signals given off from the things we need for survival and reproduction. As we go through each sense, we will see that only the most important cues about the environment contribute to our internal models. We can only see a trivial portion of the existing electromagnetic spectrum, hear a sliver of the frequencies that exist, and only smell a minority of the chemicals that exist. We are not in direct contact with the external world. Slide 17. And this is a good thing. Imagine the chaos of being able to hear every available frequency that surrounds you. You'd be able to hear every radio station, cell phone call, and dog whimper in the surrounding environment. Let me briefly lay out Plato's allegory about the prisoner in the cave and invite you into this thought experiment about how Plato saw perceptions of sense data. So, imagine a prisoner is in a cave whose only contact with the outside world is the shadows cast on the inside of the cave by the objects in the outside world. For Plato, we are the prisoner. The shadows of the outside world are our perceptions of reality, and the objects and events happening outside the cave are reality. That is, we are indirectly in contact with the external world. I hope that didn't hurt the brains of any philosophers out there. That was a super simplistic application of Plato's cave allegory, and there's much more to unpack here philosophically. But that's beyond the scope of this class. What is within the scope of this class is planting a seed of doubt about our relationship with reality without rendering it completely useless. I also like the image on the right-hand side here where it's kind of a takeoff of the cave allegory, except instead of the percepts being shadows on a wall, the percepts are images on electronic devices. I like that. It's a little social commentary a la Plato's cave. Slide 18. Let's begin with the very instruments we use to sense stimuli: the eyes, ears, skin, nose, and tongue, with an honorable mention to the vestibular system. These sensing instruments of ours are the boundary between the external world and the internal models we build about the external world. They give rise to seeing, hearing, touching, smelling, tasting, and spatial orientation or balance. Let's take these one by one. Slide 19. We'll be spending more time reviewing vision than the other senses because there has been more research on vision. This makes sense since we are such visual creatures. The sense of smell became less important the further from the ground our noses became. Also, standing upright provides a treasure of visual stimulation that isn't afforded to animals restricted to exist on all fours, so close to the earth. So what enables you to see something? Do you see because light enters your eyes, or do you see by sending out sight rays? The correct answer is that light enters your eyes. I can't believe that I have to say that, but apparently the idea that we see by sending out something like sight rays is pervasive enough that the textbook author deemed this point important enough to include it in the chapter. I had to research this, and it appears that indeed a scary amount of people, including college students, think we emit rays out of our eyes to see. This has been studied, and the results are disappointing. In 2002, a group of researchers summarized research showing that large portions of adults believe that our visual perceptions are accompanied by tiny emissions from our eyes. Diagrams showing emissions emanating from the eyes were chosen 41 to 67% of the time as correct depictions of vision. If this is how you think we see, please stop thinking that. Research also shows that explaining how the eye works while also explaining how the eye doesn't work is effective in modifying this belief. I just explained how vision does not work. Allow me to explain how it does. Critiques of the sight ray theory date back about 1,000 years ago. The Islamic scholar, and I'm going to ruin this, Ibn al-Haytham, reasoned that people see the stars as soon as they open their eyes, and it is implausible that sight rays would travel the distance to the stars and back that fast. So what is it that we're seeing? Let's start with the stimulus. The stimulus of vision is the electromagnetic spectrum of light waves. For the spectrum that is available to us, light waves bounce off objects around us, and we detect those reflected light waves. Since our main source of light, the sun, is a star, we can also see light coming directly from other light sources or stars during the evening hours. Slide 20. When we see something, light reflected from the object passes through the pupil, an adjustable opening that widens and narrows to control the amount of light entering the eye. The iris, or the colored structure on the surface of the eye surrounding the pupil, is what we describe when we say someone has brown, green, or blue eyes. Light passing through the pupil travels through the vitreous humor, which is a clear jelly-like substance, to strike the retina. The retina is a layer of visual receptors covering the back surface of the eyeball. The cornea and the lens focus light on the retina. The cornea, a rigid transparent structure on the surface of the eyeball, always focuses light in the same way. The lens, a flexible structure that varies in thickness, enables accommodations of the lens. That is, you can adjust its focus for objects at different distances. The fovea, the central area of the human retina, is adapted for detailed vision. Of all retinal areas, the fovea has the greatest density of receptors. Also, more of the cerebral cortex is devoted to analyzing input from the fovea than input from other areas. The retina's two types of visual receptors are cones and rods. Cones, found mainly in and near the fovea, are essentially for color vision, whereas rods, more numerous toward the periphery, detect dim light. The visual receptors send impulses away from the brain toward the center of the eye, where they contact neurons called bipolar cells. The bipolar cells contact still other neurons, the ganglion cells. The axons from the ganglion cells join to form the optic nerve, which turns around and exits the eye. The blind spot is the area of the retina through which the optic nerve exits. Slide 21. And there are three theories of color vision that we're going to go over. I'd like to make it clear from the outset that these are not competing theories, but rather complementary and combine to give the most accurate explanation of color vision. Let's start with the Young-Helmholtz trichromatic theory. Color vision depends on three types of cones, each sensitive to a particular range of wavelengths. Cones transmit messages so that later cells in the visual system indicate one color, for example blue, by an increase in activity, and another color, yellow, by a decrease. The cerebral cortex compares responses from different parts of the retina to determine color experiences. So let's talk about how this kind of came into be. So Hermann von Helmholtz, obviously from Berlin, New Jersey, Germany, same difference, helped us understand color vision back in the mid-1800s by tackling a color vision problem that had existed since Isaac Newton's discovery that white sunlight was a mixture of all visible colors. The puzzle has two arms. On one hand, we can generate white light by combining lights of all colors. This confirms Newton's theory. However, you can also generate white light with two complementary colors. What gives? But wait, there's more. We can also generate certain colors like orange from pure orange light. However, we can generate orange by mixing red and yellow light. Again, what gives? When you combine two complementary colors, you get white light. When you combine two colors that aren't necessarily complementary, you get a third color. Helmholtz compiled evidence, like how we perceive color from mixing lights of different hues and the existence of color blindness, to move forward a theory inspired by his research on how all perceived colors are derived from three primary colors. His tests were to have participants match a target color by combining either two or three basic wavelengths. He found that, while participants had difficulty color matching with two wavelengths, they could match any color with three. From these converging pieces of evidence, he posited that we have three kinds of receptors in our retina, each sensitive to one of the three primary colors. They convey specific color information when transmitting a visual image. The trichromatic theory, as it came to be called, is also known as the Young-Helmholtz theory because English scientist Thomas Young came up with a similar theory at around the same time. Slide 22. The next theory is the opponent process theory, and the description I'm about to give you is a synopsis of one given from the website verywellmind.com. While the trichromatic theory makes clear some of the processes involved in how we see color, it doesn't explain all aspects of color vision. The opponent process theory of color vision was developed by Ewald Hering, a contemporary and fierce rival of Helmholtz, who noted that there are some color combinations that people simply never see. For example, while we often see greenish blue or bluish reds, we do not see reddish greens or yellowish blues. Opponent process theory suggests that color perception is controlled by the activity of two opponent systems, a blue-yellow mechanism and a red-green mechanism. During their lifetimes, Helmholtz, Hering, and other respective camps argued over which of their theories is correct. It turns out both theories are needed to account for the complexity of color vision. The trichromatic theory explains how the three types of cones detect different light wavelengths, while opponent process theory explains how the cones connect to the ganglion cells. These ganglion cells are where the opposing elements inhibit each other to determine how color is perceived. A demonstration of the opponent process theory is the phenomenon of negative afterimages. This is the experience of one color after the removal of another. So here's a quick example. Slide 23. So sometimes this works and sometimes it doesn't. So just stare at this image. It is an image, obviously, of the American flag. It is using the exact opposite colors as the opponent process theory would predict. So if you stare at this long enough, stare at it, stare at it, stare at it. When I switch to slide 24, bam, you should see a negative after image of the American flag in the appropriate colors. And if this doesn't work, feel free to, find a print version of this and do it in real life while staring at a white wall. It works really well. It's just for some reason, sometimes it works in a slideshow presentation and other times it doesn't. All right, slide 25. The third theory of color vision is the Retinex theory. So in the late 1950s, Edwin Land noticed that color constancy experiments showed that color perception does not correlate with receptor responses, right? So what is perceived does not correlate with the way the receptors are behaving. That is, he was able to produce the perception of certain colors without activating their corresponding cone. Even more specifically, he found that he could produce the perception of green and blue using only red wavelengths. This became known as the Land effect. To explain the Land effect, Land proposed that both the retina and the cortex were doing some heavy lifting in both color perception and brightness contrast. The cortex was taking the information provided by the retina and processing it relative to the color and brightness of objects around it. That is why the same absolute color can be perceived as lighter or darker or existent when it is not, based on its surrounding context. Land coined the term Retinex, which is the contraction of retina and cortex, to identify the spatial image processing responsible for color constancy. So if we revisit this illusion from the beginning of the lecture, the Retinex theory is what accounts for it. Even though square A looks darker than square B, it's because square B looks like a light square in a shadow. When our brains, specifically our cortex, takes into account all the surrounding environment, that's what makes that illusion hold, that A is darker than B. But when we look at the actual spectral analysis of the colors, A and B are the same color. Our brain is being fooled. Slide 26. As mentioned before, all three of these theories are needed to explain color vision. The process of how the visual system converts wavelengths of light into a perception of color begins with three kinds of cones, each sensitive to a particular range of light wavelengths. Later, cells in the visual path code this wavelength information in terms of pairs of opposites, red versus green, yellow versus blue, and white versus black. Finally, cells in the cerebral cortex compare the input from various parts of the visual field to synthesize a color experience. Color blindness is a term used for those who have problems perceiving colors. The most common type of color blindness makes it hard to tell the difference between red and green. Another type makes it hard to tell the difference between blue and yellow. People who are completely colorblind don't see color at all, but this is really uncommon. Slide 27. Moving on to hearing, also evolutionarily important because hearing allows us to receive information from long distances, giving us an advantage understanding our surroundings. All sound begins with waves of pressure, and we've taken this sense to amazing heights with our ability to compose music, but that's a topic for another class, which I hope you take, which I hope they offer for me to teach. And when they do, I hope you take. Slide 28. The stimulus for hearing are sound waves, and sound waves are just vibrations of the air, water, or any other medium. These sound waves vary in frequency and amplitude. Slide 29. The frequency of a sound wave is the number of cycles or vibrations per second, designated as hertz or Hz. Pitch is a perception closely related to frequency. We perceive a high-frequency sound wave as high-pitched and a low-frequency sound wave as low-pitched. Slide 30. Loudness is a perception of the intensity of sound waves. Other things being equal, the greater the amplitude of a sound, the louder it sounds. In addition to amplitude and pitch, sounds vary in timbre, which refers to tone complexity. And in case that's confusing, just think you can hear the same note played on a bunch of different instruments. What lets you know that that same note is being played on different instruments is that the different instruments have a different timbre. Slide 31. The ear converts relatively weak sound waves into more intense waves of pressure in the fluid-filled canals of the snail-shaped organ called the cochlea, which contains the receptors for hearing. When sound waves strike the eardrum, they cause it to vibrate. The eardrum connects to three tiny bones, the hammer, anvil, and stirrup. These are the three tiniest bones in the human body. As the weak vibrations of the large eardrum travel through these bones, they transform into stronger vibrations of the much smaller stirrup. The stirrup, in turn, transmits the vibrations to the fluid-filled cochlea, where the vibrations displace hair cells along the basilar membrane in the cochlea. These hair cells, which act like touch receptors on the skin, connect to neurons whose axons form the auditory nerve. The auditory nerve transmits the impulses to the brain areas responsible for hearing. Slide 32. Here is just another picture of the cochlea with the individual hairs and the frequencies that they are sensitive to laid out, as well as a picture to let you know where the auditory cortex is. This is where the auditory nerve is sending its information. Slide 33. Just as with vision, we'll cover three theories about how we perceive sound, with the understanding that they are complementary and not competing. That is, all three of these principles are needed to explain auditory perception. Let's start with the place principle. So, you guys remember Helmholtz and his contribution to color perception theory, the trichromatic theory? Yeah, he was kind of brilliant and also easily bored, so he didn't limit himself to research on just one sense. Helmholtz was the first to propose that the cochlea had individual hair fibers responsible for detecting each pitch and transmitting the information to the brain. That is, pitches are perceived based on the activation of hair cells in specific areas or places along the cochlea, with high frequencies stimulating the hair cells near the stirrup end of the cochlea and low frequencies stimulating the hair cells further along the membrane. Slide 34. Further research shows that all hair cells are stimulated by any sound, providing a challenge to the place principle. As its name suggests, the frequency principle is the concept that a sound wave, through the fluid of the cochlea, vibrates all the hair cells, producing action potentials in synchrony with the sound wave frequency, sending a signal to the brain via neurons firing at a rate that mimics the frequency of the sound. That is, when, say, a 100 Hz frequency is perceived, it is because the signals being sent to the brain are nerve impulses in bursts of 100 cycles per second, the same frequency as the sound wave. Slide 35. The frequency principle was good for explaining perception of low frequencies, but ran into limitations when used to explain more intermediate frequencies. Our neurons can't fire as fast as the frequencies of intermediate and high sound waves, so how does the frequency principle apply to these higher frequencies? The Volley principle is a modification of the frequency principle that fixes this limitation by showing that at higher frequencies, a cluster of neurons can fire in rapid succession to produce a volley of impulses that are later combined to equal the perceived frequency. It turns out this volley principle only works up to frequencies of about 1,000 hertz. But fear not, even though these principles have their limitations, together they explain the range of sounds that we perceive. That is, we hear pitch by different mechanisms at different frequencies. At low frequencies of sound, below 1,000 hertz, we identify pitch by the frequency of vibrations of hair cells in our ears and volleys of responses from clusters of neurons. At high frequencies, above 5,000 hertz, we identify pitch by the location or place where the hair cell vibrates on the cochlear membrane. For frequencies between 1,000 and 5,000 hertz, we use a combination of all principles. Slide 36. When these perceived frequencies are organized according to our musical scale, we get musical pitches. An inability to recognize or reproduce these pitches, specifically musical pitches, is called amusia. Slide 37. While we're talking about hearing and the anatomy of the ear, it's worth briefly visiting the vestibular system. The vestibular system tells us about the movement of the head and its position with respect to gravity. It plays a key role in posture and balance. Intense vestibular sensations are responsible for what we perceive as motion sickness. The vestibular system also enables us to keep our eyes fixated on an object while the rest of the body is in motion. When you walk down the street, you can keep your eyes fixated on a street sign even though your head is bobbing up and down. The vestibular sense detects head movements and compensates with eye movements. Slide 38. The remaining three senses, touch, smell, and taste, will be reviewed very quickly, not because they aren't important, but because they are less researched than vision and hearing. And the theories of perception that we're going to be covering were developed with an eye and ear towards vision and hearing. See what I did there? Dad joke. Slide 39. The cutaneous system and senses. All right, cutaneous is just a fancy way of saying of the skin, and we experience many types of sensation on the skin, each dependent on different receptors. Itch is a sensation based on tissue irritation inhibited by pain. Tickle depends on the unpredictability of a stimulus. And for the purposes of this class, we'll be focusing on the sensation of pain after we talk about the skin a little bit. The cutaneous system is sensitive to temperature, pressure, texture, and irritation, among other things. The skin consists of three layers. There's the epidermis, the dermis, and the hypodermis or subcutaneous tissue. The epidermis is made up of cells that produce pigment and protect the immune system, while the dermis contains nerve endings, oil, sweat glands, and hair follicles. The subcutaneous tissue is made up of fat, connective tissue, and larger blood vessels. Since the dermis is where the nerve endings live, it is the layer responsible for sending signals of touch and pain to the brain. Slide 40. The experience of pain is a mixture of bodily sensation and emotional reaction, which depends on different brain areas. An area in the parietal cortex responds to the sensation itself. The brain area responsive to the emotional aspect is called the anterior cingulate cortex. While physical pain can be intense at the moment, emotional pain is more enduring. The gate theory of pain is the idea that pain messages must pass through a gate, presumably in the spinal cord, that can block the message. That is, other kinds of input close the gate, preventing pain messages from reaching the brain. If you injure yourself, rubbing the surrounding skin sends inhibitory messages to the spinal cord, closing the pain gates. Pleasant or distracting events also send inhibitory messages. In contrast, a barrage of pain stimuli cause pain at the time and increase response to similar stimuli in the future. One way to decrease pain is through distraction. Post-surgery patients in a room with a pleasant view complain less about pain, take less pain-killing medicine, and recover faster than do patients in a windowless room. Endorphins are neurotransmitters that weaken pain sensations. Morphine, which stimulates endorphin synapses, has long been known for its ability to inhibit dull, lingering pains. Pleasant experiences, such as sexual activity or thrilling music, also release endorphins. Another method of decreasing pain begins by inducing it. The chemical capsaicin stimulates receptors that respond to painful heat. Rubbing capsaicin on the skin produces a temporary burning sensation. As it subsides, the skin loses some of its pain sensitivity. Several skin creams with capsaicin are used to relieve aching muscles. But pain can also exist in a part of the body that doesn't exist anymore. This is called phantom limb syndrome. After an amputation, the corresponding portion of the somatosensory cortex stops receiving its normal input. Axons from neighboring cortical areas excite the silenced areas of the cortex, and when these cortical areas receive the new input, they react in the old way, producing a phantom sensation. Slide 41. And now let's cover the chemical senses. So both smell and taste are considered chemical senses because they require chemicals to bind with receptors, which trigger a signal to the brain. Slide 42. The sense of taste, which detects chemicals on the tongue, serves just one function. It governs eating and drinking. The taste receptors are in the taste buds, located in the folds on the surface of the tongue, mainly along the edge of the tongue in adults. Children, on the other hand, have taste buds that are more widely distributed. People have receptors sensitive to sweet, sour, salty, bitter, and umami, which is also a savory glutamate response. Slide 43. This image is just to demonstrate that regardless of what you hear about certain parts of the tongue being responsible for certain tastes, it is the case that the taste buds that we have for the tastes that we perceive are all pretty much all over the tongue. So, in the same way that if you thought we saw by shooting sight rays out of our eyes, I would like you to stop thinking that, like I would like you to stop thinking about the tongue having specific areas responsible for specific tastes. It's just not true. Slide 44. The olfactory system, the sense of smell, depends on hundreds of types of receptors that live on the ceiling of your sinuses. Odors are either inhaled through the nose orthonasally or travel up the back of the throat retronasally to reach these receptors, which converge at the olfactory bulb, which sends signals to the cortex. Through combinations of the responses of our hundreds of types of receptors, we can distinguish among a huge numbers of odors and their mixtures. Slide 45. We have strong emotional reactions to many odors based on previous experiences, known as the Proust effect. For example, while most would think of roses as a pleasant smell, we've had participants report hating the smell of roses, one specifically because she first smelled them at her mother's funeral, so it reminds her of that. Some would say these emotional associations with fragrances is what drives effects claimed about many aromatherapy products. Some would say that. Some. Okay, I'm saying that. I'm saying that aromatherapy is junk. Olfaction serves important social functions in most non-human animals. In many species, individuals use olfaction to recognize one another and to identify when a female is sexually receptive. The smell of a sweaty woman, especially a woman near her time of ovulation, causes a heterosexual man to increase his testosterone secretion. Slide 46. Some people have 3 times as many taste buds on the tongue as other people do. Those with more taste buds experience stronger tastes, tend to have stronger food likes and dislikes than other people, and generally dislike strong-tasting or highly spiced foods. People also vary in olfaction. We have hundreds of types of olfactory receptors, each controlled by its own gene. Many of these genes differ slightly from one person to another, and as a result, a particular odor might seem stronger or weaker to you than to somebody else. People vary in olfaction for other reasons too. On average, women are more sensitive to smells than men are, and young people are more sensitive than older people. So an example of odors smelling different to different people, some people love the smell of cilantro, and some people hate it. It's just one of those things. Some people can smell from their urine when they've eaten asparagus. Some people can't. Some people produce the smell in their urine after they've eaten asparagus, but they can't detect it themselves, and other people can. So smell is interesting. Slide 47. Ever get a cold that makes you congested? And when you go to eat something, it doesn't seem to taste the same? Well, this is actually because flavor perception, requires information from both taste and smell, with smell doing most of the heavy lifting. Normally when we eat, odor molecules are released from the food we are eating before it enters our mouth, while we're chewing it, and after we swallow it. Before we put the food in our mouth, the odor molecules are traveling orthonasally to our smell receptors, and after we place the food in our mouth, the odor molecules travel up the back of our throat retronasally. What's happening when you are congested is that odor molecules from the food you are eating are being blocked from reaching your smell receptors, particularly retronasally. So your flavor experience is missing its main ingredient, smell. All of this to say, when you have a cold that congests you, your food tastes the same because your taste buds are working just fine. You're just having a jacked up flavor experience because taste information is in fact the only part of the food your brain is perceiving. Without all of that olfactory delight, you are just getting taste with no flavor. Slide 48. Problems with smell perception come in many forms, from a diminished sense of smell, hyposmia, to a complete loss of smell, anosmia. When your receptors are sending wrong information, you could suffer parosmia, a distorted sense of smell, or phantasmia, which is perceiving an odor when no source is present. This is also known as an olfactory hallucination. Slide 49. And some people have consistent experiences of one sensation evoked by another. For example, they might experience particular letters or numbers as having a color that other people don't see, such as seeing the letter E as green or red. Or they might be able to see sounds. They might be able to hear tastes. They might be able to have an input from one sense modality and have it trigger a perception in a different sense modality. This synthesis of different perceptions is known as synesthesia. Slide 50. We have now covered the sensation part of sensation and perception. Let's now go over some super fun theories of perception. Many examples will be given. This is fun. This is a good way to end this up. Okay, slide 51. Let's revisit the point I was making at the beginning of the lecture. We are not in direct contact with the external world. After reviewing all of our sense instruments, you should now have a pretty good understanding of how limited a slice of reality is available to us. A tiny percentage of the existing electromagnetic spectrum, pressure wave continuum, or chemicals. Imagine waking up tomorrow and being able to sense all of this. Hearing cell phone signals, seeing ultraviolet rays, hearing electrons bounce off surfaces, smelling every chemical that makes up our atmosphere. We would go insane, right? Evolution has selected which portion of existing reality is actually available to us, and, to put it bluntly, it's the things that helped in our survival and reproduction, as is the case with every organism as they adapt to their environment. Even with this limited portion of reality that is available to us, we aren't perfectly detecting everything that exists in that. Well then, what do we perceive? The answer is cues. The stimuli we're reviewing for each sense instrument is the cue for that specific sense modality. For instance, chemical cues for smell and taste, electromagnetic waves for sight, etc. And from these cues, we construct an internal model of the external world. Slide 52. Simple enough, right? Now, here's the fun part. We build these internal models of the external world even if we only have very subtle cues. Also, we're frequently working with more than one internal model, and this is revealed when presented with ambiguous cues. And when the cues are downright deceptive instead of ambiguous, our models end up being completely wrong, causing illusions. Let's spend a little time unpacking these. Let's start with that first point. The internal models we carry around about the external world are based on the active process of seeking cues in the environment and requires the most subtle of cues, with everything else being filled in by the mind. To demonstrate this, I have these pictures and shapes. Look in the upper left hand of this slide. What does that look like? A cow? Maybe a puppy? Maybe a pig? It's really just 3 circles and some leafy looking things, right? There's nothing about that looks like any of the animals that we think it looks like. but those tiny little subtle cues brings to mind the perception of something that it's not. Same thing happens with the smiley face that we keep drawing. Who do you know that actually has a face that looks like that? I hope nobody. But from these subtle cues of a circle with 2 dots on top and some curvy line on the bottom, our brain perceives that as a face. And then, of course, the classic example is the one that I gave at the beginning of the lecture with the Dalmatian. Even with those subtle cues, right? If we were to input that image to a computer, there would not be enough information for that computer to figure out that there is a dog in that picture. Yet we are able to detect that. That's interesting. So this is a demonstration that we only need subtle cues to start to build internal models. Slide 53. Our next point is that we carry around multiple internal models of the external world that are reflexively triggered by ambiguous cues, confusing our attempts to actively seek the necessary cues to build an accurate internal model. I know that was a mouthful, but I think you got it. The classic example of this is the face vase picture, but I found an even cooler one in the upper left-hand side. Another classic example of this is the young old woman picture, where our perceptions shift between seeing one or the other as we unconsciously and actively scan for cues. The Necker cube example at the beginning of the lecture is also a nice, simple example of ambiguous cues that make our minds flip-flop between internal models of the external world. There are non-visual examples of this as well. For instance, in my world of research olfaction, there are odors that are ambiguous and require context cues to be perceived accurately. For example, butyric acid is a chemical that comes off of both Italian cheeses and sweaty feet. If you were to smell this in the Italian market, you'd likely walk towards it because it's appetitive. If you smelled this in a locker room, you'd likely walk away from it because it would cause you to avoid it. Slide 54. Finally, when it comes to building internal models of the external world based on cues, what happens when those cues are just deceptive? Well, cues can be so ambiguous so as to be deceiving, and the result of these deceiving cues is a completely wrong internal model of the external world. These are the situations that we classically call illusions, and these illusions can revolve around a variety of sensory cues. For example, the Mueller-Lyer illusion makes it seem that lines of the same length are actually different. Depending on what colors are surrounding a specific color, it could be perceived as different even if the color itself doesn't change. Further, parallel lines can look bowed when viewed through a pattern that resembles spokes in a wheel. A non-visual example of this is the Shepard tone. This auditory illusion makes it sound like a pitch is either going up or down perpetually. It sounds like an infinite loop of either ascending or descending pitch that never reaches its destination. Let's take a listen. Slide 55. Now that we understand that perceptions are constructed based on subtle cues that can be ambiguous or deceptive, and because of this, we are not in direct contact with the external world, I want to reassure everyone that it's okay. While mostly incomplete or flat out wrong, our ability to perceive and navigate through the external world is indeed sufficient for survival. Otherwise, we wouldn't be here, right? Slide 56. Now that we've talked about how we organize perception, let's review some theories of perception. Slide 57. We'll be briefly reviewing 3 different theories of perception, then unpacking one in particular, the Gestalt school. The Gestalt school of perception posits through demonstrations that we organize the external world based on built-in principles, or Gestalten, placing them on the side of nature in the nature-nurture debate. We are born with these Gestalten. Another theory of perception falls on the nurture side of the debate by claiming that early in life we learn perceptual constancies that later serve to organize perceptions. These constancies relate to size, shape, brightness, or color. For example, as we see a 6-foot tall person walking away from us, we perceive their size getting smaller, But we don't build an internal model where the person is actually getting smaller, even when they are far enough away to be no bigger than my pinky should I hold out and extend my hand. We still understand that this is a 6-foot tall person. According to proponents of this theory, this size constancy was learned early in life and allows us to navigate the world. Finally, we have those who think that we are born with perceptual modules that are primed to calculate the statistical probabilities of our environmental cues. This is a combination of nature and nurture, since we are only born with the capacity to calculate, rendered useless without input from experience. That is, neither the modules that we're born with or the learning that takes place in the environment are doing all of the heavy lifting when building internal models, but rather they're working together to organize percepts. The internal modules are triggered by external cues and incorporate past experiences of trial and error. Slide 58. In the early days of perception research, people trusted that we were bottom up, just perceiving what was placed in front of us. If you put the letter C in front of us, we perceived it as the letter C, the letter A as the letter A, the letter T as the letter T. People in the Gestalt school of perception saw a problem with this because with the same features, we can get different perceptions. So even if you're detecting the appropriate features, the function of that object will change. For instance, the symbol used in cat and hat for the letter A is the same symbol used as the letter H. What is it about the same exact symbol being used two different ways that allows us to use it in two different ways? There's something happening beyond bottom-up processing. There's something beyond the symbols that we're perceiving that are allowing us to understand those symbols. Same thing with the next example. Right? When you read it left to right, it is ABC. Up and down, it is 12, 13, 14. Well, which is it? Are we perceiving the letter B or the number 13? If your answer is both, then you can't say that we are bottom-up processing anything, because either it is a B or a 13. If we do any processing in our brain and bring that experience to our perceptions, we are engaging in top-down processing. And guess what? That is exactly what the Gestaltists want us to think. Slide 59. According to Gestalt psychology, we perceive an organized whole by identifying patterns in a top-down manner, right? These built-in Gestalten that we have cause us to see figure ground. It is built into us to notice figure from ground. It is built into us to notice continuity. And here's a fun little, this is a pictograph of the word Gestalt, including Gestalt principles. So you have closure. You're able to perceive that letter G even though there is a white line in the middle of it. So it's not really one character G. It's two characters separated by a white line, but we perceive it as the letter G. That is closure. The next letter E is really just 15 individual squares with some of them black and some of them gray, but the ones that are black are proximal to each other to make the letter E. Oh, so we perceive it as the letter E. The letter S has a stick going through it, but is it really going through it? Etc., etc. Let's review a couple of these. Slide 60. The Gestalt psychologists describe principles of how we organize perceptions into meaningful wholes, as illustrated in Figure 4.42. Proximity is the tendency to perceive objects that are close together as belonging to a group. The tendency to perceive similar items as being a group is, quite reasonably, called similarity, as shown in graphic B. When lines are interrupted, as in part C, we perceive continuation, a filling in of the gaps. You probably perceive this illustration as a rectangle covering the center of a very long hot dog, but it could just as easily be two different hot dogs, each with an end sticking outside of the green rectangle. There could be 3 hot dogs in there for all we know, 4, 5, 6, a dozen hot dogs in there. But we're just seeing the end of 2 of them, assuming that it's one continuous hot dog. When a familiar figure is interrupted, as in part D, we perceive a closure of the figure. That is, we imagine the rest of the figure to see something that is simple, symmetrical, or consistent with our past experiences. So, while we are perceiving an apple covering up a picture of Abraham Lincoln, it could just as easily be a jigsaw puzzle where that apple portion of Abraham Lincoln's head is completely missing, and it's two separate pieces that fit together, not one overlapping another. When possible, we tend to perceive what's called good figure, a simple, familiar, symmetrical figure. Many familiar objects are geometrically simple or close to it. The sun and the moon are round, tree trunks meet the ground at almost a right angle, faces in animals are nearly symmetrical, and so forth. If we can interpret something as a circle, square, or straight line, we do. In figure 4.42e, the bottom right-hand side, the part on the left could represent a red square overlapping a green one, or a green backward L overlapping a red object of almost any shape. We are powerfully drawn to the first interpretation because it includes good, regular, symmetrical objects. Moving on. Slide 61. We have gone over sensation. We have gone over perception. We are briefly going to go over how to measure perception because it's very statistical and boring, so I'm only going to spend a couple slides on it. Slide 62. Figure 4.32 presents typical results of an experiment measuring an absolute sensory threshold. The results show that no sharp dividing line distinguishes sensory stimuli that can be perceived and sensory stimuli that cannot be perceived. Right? So this is important. An absolute sensory threshold lets us know at what concentration a stimuli has to be for us to detect it. There are things that we cannot detect, no matter how concentrated they get. There are some things that we can't detect at low concentrations, but can detect them once they reach a threshold of detection. So being able to measure that is super important. How do we do that? Slide 63. Researchers define an absolute sensory threshold as the intensity at which a given individual detects a stimulus 50% of the time, right? It needs to be above chance. To determine how accurately someone detects a signal, we need to consider not only the ratio of hits to misses when the stimulus is presented, but also the ratio of false alarms to correct rejections when the stimulus is absent. So, let's quickly go over these terms. When I present a stimulus and there is a stimulus and you say there is a stimulus, that is a hit. When I present a stimulus and you say that there is not a stimulus, that is a miss. When there is not a stimulus and you say that there is, that is a false alarm. When there is not a stimulus and you say there is not a stimulus, that is a correct rejection. Okay. Now that we've reviewed that, when people are trying to detect some item, they are more likely to overlook it if it occurs rarely. For example, airport screeners can err by thinking they see a weapon that is not present, or missing one that is there. If they haven't seen a weapon in weeks, they expect not to see one, and they probably overlook a weapon even if they do see it. Another example is radiologists looking for indications of illness on X-ray scans. They might scan through hundreds of X-ray scans looking for small nodules that might indicate illness. With increasing expertise, they can find most of them, but they sometimes overlook something they hadn't expected to see. The process for capturing an absolute sensory threshold is known as signal detection theory, and this is just the mathematical field that calculates hits, misses, correct rejections, and false alarms. And an interesting part about studying perception is that there is a phenomenon called subliminal perception, where a stimulus that is not detected but still present can have an effect on behavior. So subliminal perception is when a stimulus affects your behavior and you don't recognize or detect that that stimulus is present. Slide 64. And we did it. We are done talking about sensation and perception. We have covered the senses, vision, hearing, the vestibular senses, cutaneous, pain, and the chemical senses of smell and taste. We've also gone over perception, how we organize it, what the theories are for it with a deep dive into Gestalt psychology, and then we briefly reviewed signal detection theory as a way of detecting absolute thresholds, and that is a way of measuring perception. I will see you all next chapter.